Hey everybody, and welcome to Chapter 2. We're talking about the neural basis of cognition, and I already feel like that is such a wildly ambitious title for a chapter, right? This idea, the neural basis of our thoughts and of our mental processes, that is something that we could spend, I was going to say an entire semester talking about, but probably more like an entire PhD could be dedicated just to this subject. So we're not going to, at the end of this chapter, you're not going to know everything there is to know about where thoughts come from in terms of how the brain creates them, but this chapter is designed to kind of give you an introduction to that, to kind of let you see how we know how the brain works and the methods that allow us to do that. All right, so what we're going to be focusing on is how do we know what's going on in the brain. This is going to come back to an idea we talked about in Chapter 1 called the black box. So if you remember that concept, we're going to come back to that and talk about what's called subtraction, the subtraction method, to talk about exploring the black box. And then we are going to talk about a bunch of different neuroimaging techniques or I should say neuroscience methods, because we will be looking at neuroimaging techniques, but that's only one half of it. We'll also be looking at lesion studies, which is what happens when the brain is damaged. And then finally, we'll talk a little bit about which method is best, because as you're going to see, and this is kind of a big idea when it comes to neuroscience or cognitive neuroscience, is that there's not one of these methods is not always going to be the best for what you need and what you want. It really depends on what kind of questions you're asking about behavior in the brain. That will help you decide which of these techniques you might want to use. So the videos that I have for us for today, the two up at the top, I don't think that either of these are necessarily crucial, but I do think they are entertaining and interesting, and they talk a lot about the concepts we're going to be talking about this chapter. So if you get, a break and want to watch, a 4 or 5 minute video about something that's not just me narrating stuff, then check these out. I think they'll give you some good context and some interesting kind of how do I want to phrase this? Some interesting flavor to our chapter. So the first is about Capgras delusion. Capgras delusion is the condition that some people have after they have a brain injury, where they see everybody who's familiar to them and they recognize them as imposters. So it's this interesting dissociation between emotion, memory, and attention. And we'll talk more about that at the very end of the chapter. And then we have, some of you have certainly heard this guy before, Phineas Gage. Phineas Gage was an early example of a lesion study. He's a guy who had a brain injury, and we learned a lot about his behavior afterwards, which told us maybe about what that part of the brain did, what it was responsible for, because we could see what happened to his behavior afterwards. Okay, that's enough kind of table setting. Let's get right into Chapter 2. So, and I should say, I just told you that was enough table setting, but I got one more thing I want to tell you, which is that I think that this chapter is one of the shorter chapters that we're going to be talking about. So even though it does talk about some really advanced technology, I'm not going to be demanding that you know a lot of the ins and outs of that technology, because a lot of you aren't going to be really working with that in your graduate career or in your career. I'm speaking from experience. Whenever I was in college, I didn't learn almost anything about fMRI, and then when I was in grad school, I was able to learn it and to publish some articles of research from using MRI. So what I'm saying is that don't feel like you're being cheated for me not going into the details with some of the neuroscience stuff. It'll come to you. This class just isn't always the best place to talk about it. All right, so let's get into it. So cognitive neuroscience, this is a discipline that I talked a little bit about in Chapter 1. Cognitive neuroscience, I would say, really kind of came to the fore in the 1990s. The 1990s was a good time in terms of research, because I'm not being political, but a lot of research about the brain is incredibly expensive, way more expensive than what an average person can afford. We're talking, for one person, tens of thousands of dollars to scan one brain in some cases. So for that reason, people need funding. And usually where scientists get their funding is from the government. And you may know that in the 1990s, there was a government surplus. Like they had a lot of cash to go around in the economy. And so a lot of neuroscience was really well funded in that era. And I think that's part of the reason why we get cognitive neuroscience. We get lots of places are getting MRI machines or PET machines and these new devices that allow them to see into the brain and what's going on in the brain. And it's an interdisciplinary field, which means that most psychologists can't just turn on an MRI machine and start scanning. Likewise, neuroscientists aren't necessarily well equipped to understand behavior. They understand the brain really well, but maybe not the behavior that results from that brain. So, cognitive neuroscience, as the name suggests, is kind of a marriage between cognitive psychology and neuroscience. And so usually papers that come out that are published in cognitive neuroscience usually have a dozen authors because you have people in really specialized fields. You have some people who are physicists, some people who are engineers, some people who are physicians, some people who are neuroscientists, and then some people who are psychologists. So it's it requires a lot of people to kind of get this stuff together and to get it done. So usually a cognitive neuroscience task is you have a participant come in, they do some kind of behavioral task while they're being measured with a neuroimaging device like MRI or PET or EEG, and then we will look at the resulting brain activity, and we will try to draw comparisons or make inferences about this area of the brain is responsible for this kind of behavior, and they interact. And that's a really short, simplified way of discussing it. We'll talk in more detail about what that means, but that's the simple idea. That you have people come in, you have them either look at something or do something or hear something while they're in a scanner having their brain scan, and then we draw conclusions about about what areas of the brain do what after looking at that activity. So, you may remember that we talked about information theory back in chapter 1, and we talked about how the brain is roughly analogous to a machine that has input and output. So we know that stuff comes into our bodies, and we know things come out of our bodies in terms of our behavior, that is our output. So what happens in between? This is what cognitive neuroscience is very interested in understanding. So information, in that way, information is going to be represented by neurons firing throughout the brain. So whenever we talk about, and you're going to hear me use this phrase information a lot throughout the class, where I talk about that there's information going from one part of the brain to the other part of the brain. What does that mean? Basically what we're saying here is that the way that neurons fire in one location is different or is changing as it goes to another location, and that's what we mean by information. Information being just kind of a shortcut way of describing that an action potential. If you remember action potentials from your Psych 101 class when you talk about the anatomy of the neuron, that information is those action potentials firing down the length of an axon or down the length of a neuron. If all that stuff is unfamiliar to you, definitely crack open Wikipedia or something like that to remind yourself. We'll talk about some of those things, but that's the basis of our conversations today. So in terms of input, we're taking these sensory organs like our eyes or our nose or our ears, and that information is going to be picking up, those pieces of our anatomy are going to be picking up information around us. That's then going to go to different parts of the brain. If I hear something, that's going to go to my auditory cortex. If I see something, that's going to go to my occipital lobe. From there, once it gets to the brain, then there are going to be networks of neurons that are going to help take that information and make it travel all throughout the brain. So when we have vision, for example, vision takes place in our eyes, happens or is integrated in the back of our heads in the occipital lobe, and then it migrates forward depending on the networks that are being used. And that communication of those neurons interweave together, communicating with one another, that is our behavior. When we behave, it's because different parts of our brain are reacting. They are activated, and that causes us to do things. So how do we know this? We know this because we have spent over 100 years getting to learn about our friend, the axon. our friend, the neuron. I said axon because I was looking at this word right here. So let's talk about what the neuron does. I'm going to very quickly kind of run through this. I'm going to show you a couple of other examples of neurons over here, but this is kind of an idealized version. They don't all look like this, but this is just the simplified version that people use as a teaching example. So here we have the dendrites. The dendrites are going to be the branch-like structures you have at the beginning of a neuron. So these branch-like structures are hungry. They are looking for neurotransmitters. They can't wait until they get some neurotransmitters to absorb, so that if they get enough of the neurotransmitter they're looking for, this is very mentalistic language, by the way. It doesn't have eyes. It's not actually looking, right? But whenever these branch-like structures get enough of the neuron that they enough of the neurotransmitters that they require, they will report over to this cell body, which is sometimes called the soma. And the soma, whenever it has enough of the neurotransmitters to reach a certain threshold, it is going to depolarize. What does that mean? Depolarize means that this cell makes a decision that it is going to fire an electrical current down the length of its body. So that current is, if you take Dr. Manella's class, which is another basic processes class for behavioral neuroscience, you'll learn that what happens is that there are sodium gates that open up, and sodium ions are going to rush into the cell, and when it depolarizes and it fires that electrical signal, as soon as it does that, the neuron is like, wait a second, we gotta balance this out. There's way too much sodium in here, and so they're going to open up the potassium channels and allow potassium to come into this to balance it back out. So it's this constant kind of opening and closing of valves, or gates, I should say, in the cell that helps keep it powered. But sodium and potassium are kind of the big players here. You're not going to need to necessarily know that for this class, but it definitely will come up in other classes when you least expect it. So that electrical current, which I talked about, is called an action potential. An action potential is going to fire down the length of this axon. The axon is just a nerve fiber that goes from one end to the next. It's essentially going to be the length so that it can take you to the next neuron in the brain, or in the body, I should say. This axon is wrapped up in these sheaths, which are called the myelin sheath. They are essentially these kind of like fatty envelopes. They're made of like this fatty substance that helps kind of conduct and insulate this piece of our body to make sure that signal doesn't get lost or get corrupted or distorted in any way as it's traveling down the length of that axon. Once it gets there, once that electrical signal travels down the length of this, it's going to terminate in these buttons. Sometimes you'll hear them called the axon terminals. Sometimes you'll hear it called the terminal buttons. Either way is fine. Terminal buttons, I feel like, is a little bit more of an old school phrase. Axon terminals seems to be the more kind of more recent, modern way of talking about it. But whenever these endings get enough of those electrical current, it lets those endings know to release neurotransmitters. And those neurotransmitters are then going to move on to the next neuron, and that this gap is called the synapse. So a synapse is between a neuron. So these neurotransmitters, these dopamine, serotonin, I don't know, acetylcholine, whatever, glutamate, whatever these neurotransmitters are, they're going to move across the synapse and get to the next set of dendrites for this cell before they move on. So that is the neuron, in a quick 5-minute kind of reintroduction to it. So neurons are quote-unquote digital. What does that mean? Digital means that they are either turned on or they are turned off. I've been talking about how we can think about information in the brain, or about happenings in the brain, as information. And that's because information, as dictated by the way that geneticists use it, and physicists use it, and computer scientists use it, is represented as on or off. It's either on or it's off. There are no in-betweens. There are no it's kind of on, or there's no it's turned off a little bit, but it's mostly on. It's either on or it's off. And these neurons, when they fire a binary impulse. That action potential that they fire down the length of that axon, no matter what the cell looks like, that signal is going to be the same no matter what. It's not going to be a really intense action potential. It's not going to be a really soft action potential. It's just going to be an action potential. It's just going to flush. Sorry, I said it's just going to flush. I meant it's just going to fire. But the reason why I said flush is because when I teach intro, when I teach Psych 101, we talk about these action potentials as having an all or none principle, which means that the neuron either fires or it doesn't. There is no in between. And I compare this to a toilet flushing, that whenever you push the handle on a toilet, it doesn't matter if you hit it really hard or if you barely do it, it's going to flush, right? That once you get enough of that lever down, that the mechanic to the toilet, it opens up, the water rushes in, rushes out, and that happens. So that's why I was saying that the neuron flushes, is because it's either turned on or it's turned off. So that neural impulse is going to travel from one neuron to the next, and it's going to do that internally as an action potential, and then between neurons communicate with each other using neurotransmitters. And that is ultimately how our brains work, is a really complex kind of interaction of all these different cells. And these are some examples of how different these neurons can look. So over here, what you see in this is just a selection of some of the types of neurons, but you can see they all have dendrites, they all have a cell body, they all have an axon, and they all have terminal buttons. It just depends on kind of where they are and how many they have. Like this one has a ton of dendrites. This one has terminal buttons that latch right onto a muscle, and so on. Alright, so that is the neuron. We have approximately, I say 100 billion here, that is a high estimate. It's probably somewhere between 70 and 100 billion, which is quite a range, but we have a lot. We have billions upon billions of neurons in our brain. Here are the 4 lobes of the brain. We're going to get to know each of these quite well. If you took sensation and perception with me, then you should know these pretty well already. But I'm going to kind of walk through and talk about what each of them do. We got the frontal lobe. I'm going to start with the frontal lobe. Why do I want to start with the frontal lobe? Because it is the smartest, quote unquote. Oh, I shouldn't have even said that. Forget it. I said it was the smartest. But I could see another psychologist watching this video and being like, that is not the correct way of thinking or conceptualizing a frontal lobe. Fine. What I mean about that is that frontal lobe is going to be used for a lot of behaviors that we normally think of as specific to humans. Again, this is a simplification. But what I mean by that is if you think about any kind of behavior that you think, well, a human can do this, but maybe a dog can't or a cat can't, that probably is going to be something that happens in the frontal lobe. So I'm talking about creativity, decision-making, reasoning and logic, complex computations, and stuff like that. That's going to be in the frontal lobe. I don't know if you did get a chance to look at it in the first chapter. I talked about one video. It was really, really long. It was like a 50-minute video, but it was about transcranial magnetic stimulation. And I said, this is a demonstration of walking through what happened, how to set up a client for TMS. And that guy talks a lot about the DLPFC, and what he's referring to is the dorsal lateral prefrontal cortex, which is right around here. This is a very crucial area for a lot of kind of behaviors that humans have issues with, whether that be problems with anxiety or attention or depression or whatever. The DLPFC, the dorsal lateral prefrontal cortex, is going to be one of those areas. And so again, think about humans. Do humans have issues with anxiety and depression and whatever? Yes. Do ants? Probably not, so anything that you think, oh, well, humans can do this, but maybe not ants, that is something that probably is going to be handled by the frontal lobe, the more kind of complex cognition. Let's move a little bit clockwise now. So we're moving from the frontal lobe over to the parietal lobe. The parietal lobe is going to be used for a lot of spatial knowledge, spatial processing, knowing the layout of a room, for example, or knowing how far it takes to get to your car from where you are sitting right now. Or even if you were poked somewhere on your body, knowing where exactly on your body that takes place, that is the parietal lobe. The parietal lobe is also going to be used in some math computation, but it's kind of used for a lot of different things. But usually space or spatial awareness is usually kind of one of the big things that people talk about. Moving clockwise still over to the occipital lobe. The occipital lobe, I shouted it out earlier today. The occipital lobe is in the very back of the head. It is responsible for visual processing. That's pretty much it, just visual processing. And if we keep moving, we're going to ultimately get to the last lobe, which is the temporal lobe right here. The temporal lobe is going to be used for many different things, but mostly speech and hearing. So you're going to have a lot of speech, language, hearing around here, because this is where we have the auditory cortex. The auditory cortex, called the A1, is going to be where we process hearing. And so we're going to see things like right around here we have a speech center. Right around here we have an area that helps us detect familiar words and phrases. And then if we move a little bit further back, we're going to see like some of these areas help us just identify objects, visual or otherwise. So the temporal lobe is going to be used a lot for language and hearing, but it's also going to be used for identification and some memory. Those are the four. Now, you may, I was very consciously making an effort to go clockwise like this. That's going from FPOT, because that spells an acronym, which is FPOT. FPOT, 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 FPOT. That is a quick way to remember the different lobes of the brain. If ever you forget, remember FPOT. Your brain is important, so FPOT. Get it? Because marijuana is not good for your short-term memory. Because that's why, alright, you get it, right? So hopefully it's memorable and it'll help you remember that you're like, oh, I forgot, what is that other, there's one lobe I'm missing, I don't remember what it starts with. Remember FPOT, frontal, parietal, occipital, temporal. So, frontal lobe. As I mentioned, frontal lobe going to be used for planning. I didn't use planning as an example, but planning, higher level processing. It's going to be used there. The occipital lobe is going to be for vision, parietal lobe is going to be for spatial processing, and temporal lobe is going to be for recognition of objects and also language and audition and hearing. There are a couple of other structures that I want to shout out. These are subcortical structures. What does it mean to be a subcortical structure? It just means that it's not on the surface of the brain. So these are a little bit more internal. They're more medial, as a neuroscientist would say. And the ones that we're going to be talking a lot about this semester, first we got the cingulate. The cingulate is this section right here. Sometimes you'll hear it called the anterior cingulate. And that's going to be used a lot in Chapter 12 when we talk about decision making. But you may have talked about it in abnormal psychology, because it comes up a lot in discussions of schizophrenia as well. Let's see, what else we got? Fornix. Don't worry about fornix. We're not going to talk about fornix all that much. The thalamus and the hypothalamus. These two structures are going to be used a lot in sensory processing. They're going to be kind of like the sensory gateway for incoming sensory signals and how they are processed before being distributed to other parts of the brain. The amygdala, this is quite a large amygdala for this model. The amygdala is going to be the emotional seat, or the emotional center, as most people kind of put it. I'm wrestling with this a little bit, because the amygdala is crucial for emotion processing, but it's not the only player. There are lots of other areas of the brain that help us regulate emotions, but the amygdala is going to be used a lot for disgust and hate and dislike. The amygdala is. And so when we have people that have post-traumatic stress disorder, for example, part of the reason why people have issues kind of controlling emotional reactions to specific stimuli is because the amygdala is kind of out of balance. Again, very mentalistic way of phrasing this, because the amygdala is not responding to incoming stimuli like it used to, and is causing some behavioral issues as a result. And then finally we have the hippocampus. The hippocampus, we're going to be talking a lot about this hippocampus right here. This thing looks kind of like a seahorse. That's what it's named after in Latin. But the hippocampus is used for memory. It's used for memory, and specifically encoding and consolidating memories. Alright, so let's take a quick look at how we know what the brain does. And I'm going to give you an example using fMRI. We'll talk more about what fMRI is later, but I just wanted to give you an example of this before we start dissecting what each of these individual neuroscience techniques are. So let's say, and this is a very basic question, let's say that we want to know what part of the brain is responsible for controlling our hands. So we already know this. This is settled science. There's no people that are questioning which part of the brain, is responsible for hand movement. So we're going to design a quick replication to try to see if we can figure it out for ourselves. Let's say that we have access to an MRI scanner. An MRI scanner means that we can indirectly measure blood flow throughout the brain, and the idea, the reason, the rationale behind this is that if a part of your body is active, it's going to be recruiting a lot of blood. Why? Because your muscles, your organs need oxygen to survive, and that's what blood is good for. Blood is good for transporting oxygen. You probably learned about that in middle school, right? And so what this device does, an MRI does, is it kind of measures roughly where there is blood. And we can conclude if there's a lot of blood, it's because that area of the body is being used a lot. Does that make sense? So if you go to the gym, you start doing a bunch of curls, you're going to have a lot of blood be recruited for your bicep, right? Because you are using that part of your body. The same is true for the brain. If we're using a part of our brain a lot, it's going to recruit more oxygen in other areas. So we can gather that area of the brain must be used more than other areas because it's recruiting more oxygen. It's recruiting more blood flow. That is the idea. All right, so let's see if we can find out how we might go about trying to figure out what part of the brain, which part of this brain is responsible for controlling a hand. So we're going to put somebody into a scanner and we're going to measure their brain. So let's say that we measured their brain. Our first instruction is that we're going to ask them to sit still, to lay down, to look forward, and to not pay any attention to anything specifically. Just kind of let your mind wander. Just kind of let thoughts come to you. So whenever you're doing this, you're going to have, naturally, your brain is going to be active and is going to be spontaneously active in different areas because your brain is always on. It doesn't turn off, and those neurons are always firing regardless of what you're thinking about. So here we have somebody just lying still and looking ahead. So we got some activation throughout their brain. Cool. Now, the next instruction is I may say, I want you to wiggle your right hand, and now I'm going to measure what's going on in the brain. So again, we see these same three areas that are active, just like before, but now we have a new area, this area right here. Now we might suspect, okay, it looks like this area of the brain must be responsible for hand movement, but we can't actually be 100% certain. What we need to do is to use the scientific method here. This is where we can conduct a little bit of research and try a quick experiment. We got this area, now what would you want to do next? What I would want to do is I would want to go back to the first instruction. Now just lie still and let's record what goes on in your brain. And so when we do that, that area goes away. That is making me feel pretty confident now that this area right here must be for hand movement because it wasn't active when they weren't moving their hand. It was active when they were moving their hand, and then when they stopped, it was no longer active, right? But maybe it's just shy, maybe it's related to something else, maybe that area is active no matter what part of our body moves. We need to control for that. So maybe our next instruction will be, we want you to wiggle your right foot now. And now you can see that this area right here is active, right? So it wasn't the same area right here, which we were suspecting might be for our hand movement. So then we would probably ask them to lie still and look ahead again. Okay, that area went away, which is good. We would expect that. And then we'd probably want them just to be careful, just to be sure of ourselves, to ask them to move their right hand one more time to make sure this area lit up and was activated, that it was recruiting more blood than other areas. So this is how we are able to understand kind of what's happening in that black box. Think about Chapter 1. Chapter 1 was all about, or not all about, it was about the history of cognitive psychology, but a big part of it was about how we can't really know what's going on in the brain. This is an example of how technology can help us understand or get a little bit closer to being correct about what's happening in the brain while we might not be able to directly peek inside and see. What this shows us is that this area is active, this area is active when we're moving our hand, this area is active when we're not moving our hand, which means that if we subtract this activation from this activation, that what's left is this unique area that is only active when we are moving our hands. So that's why we're not going to say that this area right here and this area right here and this area right here are active whenever we are moving our hands, is because that seems to be unrelated activation. That might be activation from following directions. It may be activation from having your eyes open. It may be activation from just thinking to yourself. It doesn't matter necessarily because we have isolated the area that is only active when we are moving our hand. That is the subtraction method. The subtraction method, very simple in my opinion, very kind of simple way of analyzing things, but that's what's used. That's on these multi-million-dollar pieces of technology and equipment, we're using the subtraction method to draw conclusions about what areas of the brain do what. So, and then again, really, really simplified because the actual analysis that allows us to even find what areas of the brain are active is highly, highly, highly complex and requires really intensive computing resources to do that. For example, and I'm going to be talking about my spouse a lot this semester because she does a lot of brain research. She's a clinical psychologist. Her areas of expertise are trauma, suicidal ideation, and borderline personality disorder. And she uses PET imaging. And whenever she wants to analyze data for her projects, her university has essentially a grid of servers that she's able to log on to so that she can use those servers to help analyze the findings, and not just her computer, not just her laptop. She has to have like heavy duty computing to do that. Another reason why neuroscience is so expensive and why it relies so much on external funding, like through the government, for it to be possible. In neuroimaging, this is how we draw conclusions, is by using the subtraction method. I'm not going to go over this again because I just kind of talked about it using those images, but I do want to focus on this example right here. So let's say that you want to know what area of the brain is active whenever somebody is looking at violent images, but only violent images. You're not interested in other kinds of images, just when they are looking at violence. So the way that you would do that is very similar to what we just did, where you'd have people sit in a scanner, you're measuring their brain, then they're going to sit in a scanner while they're looking at a violent image. But it's not just enough to then do the subtraction method. Then we want to also have them looking at images that are neutral, images that don't have any violence, and then subtract that from looking at images that are violent, so that with what you're left is just the activation that is unique to when people are looking at violent images and not when they're looking at any other kind of image. So we'd be subtracting the neutral image activation from the violent image activation. Alright, so the primary sensory cortex, also another really important area of the brain, and this is the area that helps us move and that helps us have awareness of our bodies. And so you can see what they call the S1, the primary somatosensory cortex, where essentially our bodies are laid out anatomically, or roughly anatomically, across our head. So that's how we know. This is how we know what areas of the brain are responsible for detecting our fingers, or for detecting our shoulders, or for detecting our lips moving or something touching our lips, is from using MRI, or I should say neuroimaging techniques, and then using the subtraction method to isolate what areas are active. Alright, now is a really good time to take a break. In case you want to take a break, you want to grab a cheese sandwich or something like that. We're 30 minutes deep. I think we're not going to go another 30 minutes long, but so it's not exactly halfway, but we're between ideas right now. So now's a good time to take a break. I'm going to take a sip of coffee. Okay, alright, so let's get into neuroscience methods. Before we even begin, though, y'all know that I like history, right? So I could not resist. I had to talk a little bit about history. You got to be careful about phrenology. Phrenology, everyone thinks, is something that took place in the past and is like a pseudoscience, and it is, but there are still some ideas, for better and worse, that phrenology still has in culture. What is phrenology? Phrenology is this dead science. It's a pseudoscience, so it's not a real science, but it looked like a science from the 1800s and into the early 1900s. And the idea was that phrenology believed that the brain had different modules all throughout its head. And I'm just going to fast forward for a second because I'm going to show you this image right here. This image right here is an example of a phrenologist diagram where you have, let's say this part of my brain is going to be for cautiousness. So, if I were to damage that part of my brain, then I would no longer be cautious, according to phrenology. But also, if you were to feel my head and you felt like this part of my skull was a little bit thicker or a little bit protruded like a bump compared to other brains, then that must mean that I am a particularly cautious person because this area is bigger, is enlarged. Likewise, if let's say that this part of my brain right here, you felt this part of my brain and it kind of recessed in, it was smaller, it was a dent, that must mean that I have a really bad sense of time because according to them, this area of the brain controlled my perception of time. Now, this is all bullshit. This isn't actually true. I shouldn't say curse words on YouTube where people can pause it and then show other faculty members that I'm swearing on the internet. But that's how that's kind of where we are with phrenology. Phrenology isn't true. It's not a real thing. It's totally made up. But you still see some of these ideas kind of circulate. They'll come up on social media where people talk about how different parts of the brain do different things. And this is going to be kind of controversial, so I apologize. A lot of people will really talk about how they are left brain people or right brain people, or how they are blank type of learner and stuff like that. There's not really any good evidence at all on learning styles when we talk about brain stuff, what's going on in the brain. If you look at peer-reviewed literature, there's not any evidence to support. I'm a visual learner, and so my visual learning style means that I have more activation in this part of my brain. That's not how the brain works. You might be a visual learner, but that doesn't mean that your brain is organized differently than other folks. That's what I'm trying to say here, is that doesn't necessarily mean that your brain operates differently. Alright, so that's phrenology. So it's important to keep phrenology in mind as we talk about some of these ideas, because this idea about how the brain works being modular and being like independent from one another is not true. That the brain is highly interactive, that it interacts with itself all the time, and it's very, 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 very rare to have an area like the amygdala be the emotional part of the brain, because most parts of the brain do many different things, and it depends on where it's getting its information and where that information is going to. You might have one area, for example, we were talking about the anterior cingulate cortex earlier, and somehow you might see that in abnormal psychology when we talk about schizophrenia. You'll also see it in decision making when we talk about task switching, learning to set one thing down and to pick up something else. We see it when we talk about gambling too. So we have different parts of the brain. They all do different things, and they interact a lot, so they're not completely independent like that. With that being said, there are, like I mentioned, some examples like the amygdala or the hippocampus that do have really dedicated specific functions that it doesn't deviate too much from. And that's an example of how some of the ideas of phrenology have kind of are still present in the way that we talk about the brain. All right, so let's see. We talked about that, and if you, I was about to go on a tangent about the history of phrenology, but this isn't the appropriate venue for that. Which method is best? If we're using neuroscience methods, I want to take a step back and say that there's not going to be one best way to scan a brain. That is both a good and bad thing, I think. Sometimes students don't like that because students like having one specific answer. And unfortunately, this isn't one where there is one specific best answer, that it could be really up to what you are studying and how you're studying it, what kinds of questions you have. And so the Chapter 2 discussion that I have is going to be asking you to identify a research question and then identify what you think would be the best method to study that specific question. So whenever you're reading an article about a brain, about some kind of neuroscience technique, I want you to think about, is this the best method to do this? Or is this just the method that they had available to them and they're just trying to do their best with it? All right, so let's take a quick look at the two different flavors of neuroscience. We'll be looking at lesion studies and neuroimaging. Let's look at lesion studies first. A lesion is going to be whenever your brain has experienced some damage. That damage can be temporary or it can be permanent. It can be like having a stroke, for example. If the stroke is severe, there might not be a lot of recovery for that part of the brain. It's a permanent lesion in that case. That can. I wanted to move on, but I also want to address this temporary part. When can you have a temporary lesion? If you have a concussion, for example, that might operate like a temporary lesion where you lose some access to brain function in that part of your brain temporarily, and then it comes back over time. You can also have this with less serious strokes as well, where people have a stroke, maybe a minor stroke, and regain function there. It can also result from surgical removal where part of a brain has been removed, like if you are familiar with lobotomies. A lobotomy would be an example of a lesion study. So these studies are really important, and they are kind of the first versions of neuroscience methods that we ever had. This is, before we developed MRI or PET or EEG, we were able to look at what happened to people's behavior after they experienced some kind of brain trauma, because that tells us about what that area of the brain does compared to others. Phineas Gage is a great example of this, so that's why you've probably heard his name before in psychology. But there's a video that I posted at the very beginning of this slideshow where you can click on it and you can watch an in-depth kind of, I say in-depth, it's only 4 minutes, but it's really interesting, an in-depth kind of introduction to Phineas Gage. But Phineas Gage was a guy who had a railroad spike move through his face, and this part of the prefrontal cortex, he lost a lot of his brain. It just came out of, I don't know how else to talk about this without it being disgusting, so feel free to mute me for the next 10 seconds because it's going to get nasty. Basically, this railroad spike caused him to lose part of his brain. That, this spike blew it out of his head, essentially. And because he was missing that part of his brain, we can draw conclusions about how he behaved afterwards might be because of the prefrontal cortex. So people would talk about how he would be more impulsive, how he cursed more, how he had problems regulating his emotions, and he had some personality differences. And people concluded at the time that might be because of maybe the PFC is responsible for personality and impulsivity and using good manners. The video that I posted talks about how that's not exactly that simple, but it's an interesting case. We'll talk about more other lesion studies and kind of famous people in psychology about that later. Transcranial magnetic stimulation. This is one that we talked about in Chapter 1. I had some videos for you on that. TMS is an example of a temporary lesion because you zap somebody there in that part of their brain and it only lasts to disrupt their brain function for a second, maybe even less. It's a very powerful magnetic field that is going to scramble your neuron's ability to fire a coherent action potential, or to fire a coherent electrical signal. And so TMS is essentially, if I wanted to know, what does this part of the brain do? Then I could zap that part of the brain while trying out different behaviors to see which behavior I couldn't do. And that would be an example of a lesion study. This video right here gives you an example of using that for, I think, speech, where this guy tries to say something and they target this part of his brain and he can't actually get the words out until they turn that signal off. So that's TMS. You can kind of see an example here where usually they have people's head kind of propped so that it's like in a very specific, they can be very careful about what part of the brain they're deactivating. All right. One final lesion study that I want to talk about is split brain research. this is another one of those things where I could talk about this in an entire class. I think it's fascinating. You may have heard about this before, where in the 50s and 60s, people were desperate to try to find a good cure for epilepsy. And there weren't really good medications for it, like there are now. And the way that people would go about doing it is that they would actually sever the bridge of neurons that connects your left hemisphere and your right hemisphere. As most of you probably know, we have two hemispheres, and they are connected by a piece of our anatomy called the corpus callosum. The corpus callosum is that bridge that connects these two hemispheres. Without this bridge, the two hemispheres cannot communicate. So the reason why this was done for people that had epilepsy is that if you had a grand mal seizure, if it starts in one hemisphere, by severing the corpus callosum, it can't migrate over to the other hemisphere, which means that you still have some control over your body, and that's preferable to having no control over your body. So people that had this very radical surgery, you might think that it would totally change their lives. For the most part, it didn't really affect their behavior that much. It did reduce the severity of the seizures, which was great, and that's why people kept getting the surgery. But it wasn't until many years later that people realized there was actually an interesting kind of incidental, an interesting accident that happens whenever this surgery is done. And these two hemispheres can't communicate, which means that there are some cases where you might have information entering into one hemisphere, but that we can't act on it or we can't say what it is because we need the other hemisphere to be able to do that. This is a fascinating video that if you don't get a chance, and I'm not gonna, well, hold on, let me think. I think the Phineas Gage video is a good idea to watch. I think this one, it may not come up on the exam, but I think it's fascinating. And it's a video, it's really old because there's not a ton of split brain patients still alive today. But you can kind of see an individual who has split brain, these two hemispheres that don't communicate with one another, and you can see how his behavior is not all that different than what you would expect, except whenever you show one side of the brain something, and you show the other side of the brain something else, and then you have someone try to explain what it is they saw. All right, so in that case, and I don't want to go into too much detail here because this, again, I could spend an entire hour talking about this. You have somebody who's looking over here, and they see their left hemisphere because it's contralateral. The things that we see in our right visual field is processed by our left hemisphere. Things that we see in our left visual field are processed by our right hemisphere. So if you're looking forward, that this person can say that they saw a fork, because speech is generally in our left hemisphere. But if I ask that individual to reach over and to grab what they saw on the screen, he's telling me he saw a fork. He's not telling me he saw a spoon. But when he reaches in to identify the object that he saw, he's going to pick up a spoon because the right hemisphere saw the spoon. And that right hemisphere is controlling his left arm because it's contralateral. Again, it's the other side of the brain handles the, you've heard this before. Your left brain controls your right side of your body, and your right brain controls your left side of the body. All right, let's wrap this up by talking about the different neuroimaging techniques now. So in neuroimaging, I would say there are three big players here. There's EEG, PET, and fMRI. So the name of the game here is trying to track what areas of the brain are active and when. And by finding that, then we might be able to draw conclusions about what those areas actually do for our behavior. As you can see, my dog just let himself in, which is quite adorable. He heard me say that it was time to wrap up, and so he thought he would kind of check in on me. I'm going to pause it real quick to make sure that he doesn't need something, he doesn't need to go to the bathroom or something like that. So I'll be right back. Alright, I apologize for that. Me and the dog reached a compromise. He's hiding under the desk right now, where he likes that spot. It's like a dark little cave, and he just hides under there sometimes whenever my wife is done working in her office. Because otherwise he stays under her desk. So anyways, as I was mentioning about neuroimaging, is that is to try to help us identify what areas of the brain do what. And these are the three that are most widely used, so those are going to be the ones that we talk about. They're also the ones that you can find in your book. And I want to point out that these measures are indirect. Even though I am somebody who, I've done this research in the past, I think everybody should be much more skeptical of these findings than how we currently are. A lot of folks, and I know this is true because I teach psychology, and I see what happens if somebody's skeptical about something, and I say, oh, there's actually neuroimaging data that shows this. People then accept it as truth and accept it as gospel, and are like, oh, well, I guess it must be true because it looks like real science. It's important to know that there are some really big limitations for neuroimaging, even now, today, So these measures are indirect. That especially PET and MRI, those are indirect. They are looking for, they're actually measuring blood flow through the brain. They're not actually measuring brain activation. EEG, I will give EEG a pass. That has the most direct, I would say, because you're actually measuring electrical activation. But for PET and for MRI, you're actually just or fMRI, I should say, you're actually measuring the blood flow. And we'll talk more about that. But it's important. I feel like that's an important thing to know, that whenever somebody is showing you, a scanned brain that has different levels of activation on it, that what that's mirroring is actually the levels of blood flow throughout the brain. And we're assuming that there's more blood in those areas because those areas are being recruited or are recruiting blood and oxygen because they're being used more. All right, the three major techniques, we got EEG, PET, and fMRI. We're going to go in this order too. So EEG is going to detect electrical activity from neurons. PET is going to track a radioactive tracer that's injected into the blood, and fMRI is going to be checking oxygen levels in blood. So let's look at EEG first. EEG is short for electroencephalography, which is non-invasive, which means that you don't need to crack open someone's skull to find out what's going on. Essentially, and you've seen this in movies and television before probably, you put on like this weird shower cap and it has like these electrodes attached to it. That's EEG. It gives you data that looks like this. And what that is essentially going to be looking at combined electrical activation that's created from neurons that are firing out action potentials. So you remember action potentials, that's an electrical current. We're detecting those currents using EEG. The advantages of this is that it's fast. In other words, sometimes you'll hear this talked about as and I feel like I should have used this language because y'all are grown-ups and y'all can handle it. Temporal resolution. Temporal resolution is another way of saying that something is fast or has short temporal resolution, which means that the moments between where the brain was active and when we can record that signal is very fast. It's actually instant for EEG, or effectively instant. So it's very, very fast. You can pick up the signal very quickly, is what I should say. That doesn't mean that the experiment is short. It just means that we can pick up the activation as it is happening. It's also non-invasive, so you don't have to, crack open somebody's head like I mentioned. And you can also look at the overall brain all at once. So you have this used most commonly if somebody's doing a sleep study. The most common neuroimaging technique is going to be sleep, or is going to be an EEG. Cons, so the drawbacks to this though is that it can be kind of weak and its signal is very noisy. That's what we mean here, where you see like the signal is kind of going all over the place. It's hard to kind of interpret what we're seeing here. That's one of the downsides to EEG, is that it can be hard to interpret for that reason. Why is it so noisy? Why is that signal so noisy? It is because there are different layers of the brain. And if I'm trying to measure something that's happening in the bottom layer of the brain, I'm getting all this unnecessary activation, or that irrelevant activation from other parts of the brain. All right. We can also use it with kids. So the other techniques we don't really use with infants for a couple of different reasons. One, for PET imaging, because they deal with radiation and children are very sensitive. And for two, for fMRI, babies move way too much and you have to be completely still in a scanner. So here is an example of, you might not be able to see this because of my image and video feed on this slide, but if you download the slide, you'll be able to see there's looking at the differences in sleep stages using EEG. Let's talk about PET. So let's talk about positron emission tomography. Positron emission tomography is the most expensive of the techniques that we're discussing today, and it is whenever somebody uses a cyclotron to create a radioactive tracer. Very expensive, by the way, because there's only a few of these things on the eastern seacoast. Like, there's not that many in the world, let alone in America. Yale has one, Boston has one, Charleston, South Carolina has one, I think Pennsylvania has one, maybe a place in Florida. There's not that many that are just dedicated for research these days. But this radioactive tracer is going to be intended to bind to a specific thing. So you might create a tracer that is going to target a neurotransmitter or to target a specific part of the neuron. And then when you inject somebody, you give them 30 minutes, you're scanning them while they're doing a task, and then you can kind of see what areas of the brain that tracer is still in. That can tell you, for example, here, let's say, for example, that this was just looking at cell growth, so that they're targeting cell growth using a radioactive tracer. We can see that somebody with a normal diagnosis, that their brain, that this is the areas where cell growth is happening. For somebody that has Alzheimer's, you can see there's less cell growth overall, and that in fact there's huge areas of cell death right here in the middle. And then for somebody that has mild cognitive impairment, kind of a mix between the two. The advantages of this is that it allows you to actually measure chemical reactions happening in the brain. You're not necessarily measuring just an indirect observation. And so that is nice. That is one of the advantages. The disadvantages, though, is that, like I mentioned, it's wildly expensive. It can cost as much as $10,000 to run a single human subject here. And you can't just publish a paper with one subject on it. But also that blood flow takes a while for that tracer to kind of find its way up there and be concentrated. It takes about 30 seconds to do. And most of the thought processes and behaviors that we're interested in they're very, very fast. They're very quick, and so we need longer than 30 seconds. So this would be slow compared to EEG, which is fast. That's what we mean by that. And the fact that it deals with radiation also limits the amount of trials and participants that we can use. So if you go in for one PET study, I think you can do one more each year, and then that's it. You can't do more than that because it's too much radiation. You risk developing some kind of health issue from all the radiation. One more really quick study about my wife, because I mentioned she does PET research. She works at Yale at New Haven, and so in New Haven, actually, I don't know if I can say this, Pope. I'll say it, but you can't tell anybody though, because I'm not sure if I'm supposed to say this. When they're transporting somebody that has a radioactive tracer injected into them, they can't put them on street level and drive them around all willy-nilly. They actually have to transport them underground, so that the radiation is not going to, be picked up, by some kind of satellite. And I don't know, that somebody's not going to use a Geiger counter on the street and be like, oh my gosh, this person is about to explode or whatever. No, it's just a radioactive tracer. So they do it all underground, and there's apparently a series of tunnels I've never seen. She could be lying to me, and it could be just a fascinating urban legend. All right, and then finally we have fMRI. This is the one that I personally have most experience with. When I was in grad school, I did some of this research. So fMRI is going to be detecting changes associated with oxygen levels in the brain. You're going to see this referred to as a BOLD signal. BOLD means blood oxygen level dependent signal. So the BOLD signal that's the dependent variable that these machines pick up, is a measurement of looking at oxygen. And we're assuming that there's more oxygen in certain levels because there's more blood in certain levels, and there's more blood in certain levels because that area of the brain has been used more, and we assume that because that area is being used more, it needs more blood. So as you can see, there's a lot of inferences that are required for us to make these claims. Now, the nice things about fMRI is that it has very high spatial resolution, which is a way of saying that it can pick up on small changes, tiny changes in the brain, about 1 millimeter. So what that means is that when they do the analysis here, they're able to essentially look at 1 millimeter cubes throughout the brain. They're able to divide the brain up into 1 millimeter cubes to look at whether or not those areas are active or not. So that's much better than the others that we've talked about in this chapter. It has okay temporal resolution. Honestly, not great, because it's between 1 and 4 seconds. I see some people who are a little bit more conservative say it's about 6 seconds before that takes place, but it's faster than PET, right? It's not as fast as EEG. But what this means is that if we're measuring something like attention using fMRI, we have to be very, very careful, because attention takes place at the drop of a hat. It's very, very quick, right? Happens in less than a second for you to change your attention from one area of the room to the other. So that means that maybe attention is not so good at being studied using fMRI, because we're looking at behaviors that are thought processes that take about 1 to 4 seconds. It doesn't involve any radiation because it uses a very high-powered magnet for it to do its work. It's relatively cheap, I would say. Not as cheap as EEG, but certainly more cheap than PET imaging. And oftentimes, one of the reasons why it's cheap is because universities will partner up with hospitals, and hospitals, they may not be using their MRI machine after 5 p.m. at the end of a workday, but they can't actually turn off an MRI machine. One of the most expensive parts of it is turning it on, because it has to always stay on, because if you turn it off, you have to flush the helium out of it, and helium is becoming extremely expensive because, I don't know if you knew this, helium is a limited resource on planet Earth. It's not naturally occurring. I could be wrong about that. I think we get most of our helium from the sun over time, and we're actually running out of helium because people have used it so much. So we don't want to turn off the machine, because that means we need to get more helium, which is super expensive. So oftentimes hospitals will allow researchers from universities to come in after hours and collect their data there. And so there isn't risks about radiation or anything like that. And many of you have probably had an MRI before, where you've had your brain scanned before. The disadvantages, or maybe your knee or your elbow. So there's not really any risks. The disadvantages here is that it's only really tracking blood flow, so it's not really tracking, neurons specifically. And ongoing brain activation can be very tricky to deal with. That's why we use the subtraction method to try to reduce the ongoing brain activation. So stuff that's unrelated to the experiment, maybe somebody's thinking about what they're going to have for dinner, and we're picking that up in the scanner. So that can be a problem. And also brain differences, anatomical differences between all of our brain sizes can lead to some trickiness in understanding or interpreting our signal. So what is the best neuroscience method to use? It really depends on what you value as the researcher. So there is a time and place for all of these, to be honest with you. So let's say, for example, if you are interested in whether or not someone can see without an occipital lobe, you probably would want to remove the occipital lobe, right? So that'd probably be a surgical removal type thing. If you just wanted to know the effects of temporary blindness, then you might use transcranial magnetic stimulation, because you're going to use that signal to disrupt the occipital lobe. If you are looking at, for EEG, if you're looking at attention, if you're looking at anything with kids, you're probably going to want to use EEG. If you are looking at anything involving neurotransmitters or cell growth or cell death, I would say probably positron emission tomography is the way to go. And if you're looking at, and I would say if you're looking for a happy medium between those two, I think fMRI is a good way to go. Looking at most behaviors, I would say. Let's take a quick look at neuroimaging examples. So I'm going to give you an example using fMRI, so you can see this is a study from the 90s that is often cited because it's a very clear, kind of easy experiment, where they had people in an MRI machine and they asked them, we want you to either look at the face, that's what they told one group, or to look at the house, that's what they told the other group, or the other condition. So they get these images and they're asking you, I want you to focus on the face or I want you to focus on the house. Importantly, no matter what, your eyes are picking up the entire image, right? If I ask you to focus on on the face, you can look at the face and you can focus on that and you can identify those features, but your eyes are still picking up information from the house, right? So again, asking someone to either look at the face or look at the house. Very easy experiment. But what they found is that whenever people are looking at the face, that there is more activation in the fusiform face area, this part of the brain right here, compared to when they are looking at houses. And when people are looking at the houses, that we have, I should say that this is when they are looking at faces, and this is when they are looking at houses. So even though they're seeing the exact same image, their brain is behaving differently based on what the instructions were, which is really, really fascinating, right? This study, by the way, we'll talk more about it later in the semester when we talk about object recognition too. And so here you can even see, like using fMRI, we can track changes over time, so we can see how the parahippocampal place area, for example, how that is going to increase in activation and then decrease immediately afterwards, and how the fusiform face area, which detects faces, is going to dip and then go up really high here. So we can track these changes over time, is what I'm trying to say. All right, this is just going to give you a couple of exercises for you to kind of think about which of these might be best for your specific study. And then the final things I want to mention, because I like to try to wrap up these chapter slides by kind of starting or ending where we started. I want to talk about Capgras delusion, because Capgras delusion was one of the videos that I recommended you check out. It was a little bit longer. It was like a 9-minute video, so don't watch the whole thing. But it's about a guy who has Capgras delusion. What is Capgras delusion? It means that he recognizes his mom, but he thinks that she is an imposter that looks and sounds exactly like her, but he feels no connection to her. He doesn't feel like he loves her because it's an imposter. So why does that happen? If we were to try to resort to a behaviorist explanation, we would only be able to look at his resulting behavior and try to draw conclusions. And we might say, oh, maybe he just no longer has feelings for this individual, and that's why he feels nothing. If we were to ask a psychoanalyst or somebody who's Freudian, they'd probably have some other kind of explanation about unconscious mechanisms and subliminal, mechanisms or whatever. But using neuroscience, using cognitive neuroscience, we're able to actually figure out what's going on in the brain whenever people have Capgras syndrome. And what they found is essentially by scanning people's brains that have Capgras syndrome, they essentially find out that it's usually because of neurological damage, but because specifically the amygdala has been damaged. And what that does is it means that there's less of an emotional response. So this individual may see somebody that they recognize. The hippocampus is going to remind them, yes, I remember that person. They're very familiar to me, and yet I feel nothing. I feel no warmth, no happiness, no anger towards this individual, to this memory of this person. To this recognition of someone, I should say. The prefrontal cortex is usually damaged as well, which is used for, like I mentioned, logic and planning and stuff like that, which means that the prefrontal cortex is not able to handle some of these illogical thoughts like it would have otherwise been able to do. And that leads to Capgras delusion. All right, we went a little bit over 1 hour. I apologize for that. I tried to keep it less than an hour, but I just got too excited talking about brain stuff, as usual. So thank you so much for tuning in. We'll talk in Chapter 3 about perception, which I think is one of the most detail-oriented chapters, but we'll try to make it fun. So I will see you later. Bye-bye.